morning, everyone. It is my pleasure today to introduce our grand round speaker, JSeegmiller, who is also currently our chemistry fellow. And Jesse actually graduated from University of Minnesota Duluth with a bachelor degree in chemistry in 2000. And then he went to University of Wyoming and did his PhD in analytical chemistry. And he was able to actually finish his degree in 4 years, which is actually quite impressive because as somebody who have gone through analytical chemistry graduate program here, that I know how difficult it is. And then so most people actually would, if they could graduate in 5 years, it's fortunate. And he was actually able to do it in less than 5 years. So that shows that how productive he was and while he was in his training program. So after he graduated with a PhD in analytical chemistry, and he actually went to come back to Minnesota, where he grew up, and then he went to become a research associate at the endocrine lab in Mayo Clinic. And then he spent 1 year there and learned about mass spectrometry and also developed mass spec-based assays for many steroid hormones there. And then followed by spending 4 years in renal function lab, where he worked on developing mass spec-based assays for lohexol, lothalamate, and other molecules for evaluating renal function. And also, he also developed a gold standard method for measuring urine albumin. And then that method is still used in the renal function lab at Mayo. And from one of the talks that we heard from Dr. Lori Buckland a few weeks ago, and we know that that method is actually being used for the urine albumin standardization effort nationwide. So that really shows that how much expertise that Jesse has in developing mass spec-based method for clinical application. So, but after actually 4 years in the in the renal function lab, he actually then went to work for ABScience, which is a mass spec company where he provides excellent technical support for many clinical labs in the country to develop mass spec-based assay for clinical application. And then he still interested to become a clinical director, so that's when he decided to actually come back to academic and to actually receive formal training in clinical chemistry so he can use his expertise that in mass spectrometry for future application in clinical laboratory medicine. And that's when he started his clinical chemistry training with us, which was very fortunate for us that so today that he's going to give us a talk entitled Applying Mass Spectrometry in Laboratory Medicine and Pathology. Thank you.
All right, can everybody hear me? Thank you for that introduction, Danny. Also, thank you for accepting me into the program, and Dr. Kleen, Dr. Eckfeld, Dr. Carter for taking me in and teaching me a lot. There's a lot that I didn't know, even with a lot of background. Also, too, many of you took me into your labs, and it was probably the first time I'd ever seen certain things, bone marrow aspirates, things of that nature, and kind of walking me through and being patient with me. So it's much appreciated. I do have some disclosures before I start. Most mass spectrometry methods are not approved by the Food and Drug Administration, and definitely the methods I'm going to discuss today are not approved either. I also own Danaher stock, so as she talked about, I worked for a mass spec company before this that was owned by Danaher, but I will not directly speak to any of their products, and they'll be all generalized. Any products that I do mention are not a Danaher company. So the objectives after this talk, you should be able to recall some basic concepts of mass spectrometry, and then really recognize some challenges and opportunities within the various clinical workflows, such as small molecule and proteomics applications. And then really my goal for this talk is for you guys, and I wrote this for pathologists, to maybe identify some potential areas of integration of this technology in your work and in your research. So I didn't dive too deep into my research, but I highlighted some aspects that hopefully you can carry on to your lab and think about. So when we think about a mass spectrometer, what's a mass spectrometer actually do? Well, it measures the mass of the compound. And the way that it's able to measure the mass of the compound is it measures the mass to charge ratio. So a compound has to be charged in order to go into the mass spec. So you need potentials to pull these molecules into your mass spectrometer. And a compound can be, and this is one thing I didn't know when I went to Mayo Clinic, is a compound can be charged positive or negative in mass spectrometry. Most people don't even talk about positive things, but they can be negatively charged as well too. But if I have to one-line this for you guys, the important thing to remember about a compound is it has to be charged in order to be analyzed by a mass spec, okay? If you take nothing from my talk, take that. It has to be an ion. So here's a high-level overview of LCMS. So you have some sort of liquid delivery system, and you put your sample into the ion source. And the ion source is where the ions have to be made. So we have to be an ion when we come out of the source. So once we're an ion, we can get carried into the mass analyzer. I purposely put this as a black box. The analyzer can then separate by mass, detect the ions, and the results come out on your computer. So that's from a high-level overview. So here's the basic layout of just a mass spec. So we have the ion source where the ions are made. That's really important, right? We can't analyze it unless it's an ion. And then once the ions get in, here's the mass analyzer. This is where the mass to charge ratio is going to be filtered or measured. So I think when people go into research and they start reading literature on mass spectrometry, it's really daunting. There's a ton of different types of sources, and there's many different mass spectrometers. So I kind of listed some sources. There's electrospray source, atmospheric pressure chemical ionization, photospray, matrix-assisted laser desorption ionization, inductively coupled plasma, DART, DESI, the list continues. I could list several more. One thing I want to note is, I think this will be really of some interest to you guys, this source called rapid evaporative ionization mass spectrometry. And I'll kind of touch on that towards the end of my talk and how that can maybe apply to pathology. But clinically, in general, if you look at the lab downstairs, the drug analysis lab, primarily they're using an electrospray source. I'll talk in detail about that just to kind of give you an overview. There's also atmospheric, so with electrospray source you can analyze things like drugs, endogenous compounds, therapeutic drug monitoring, and then you go to this APCI source. So APCI is a little harsher ionization technique. So things they might analyze with that are steroids, sterols, things that don't ionize quite as well, a little harsher. And then there's inductively coupled plasma. So things that you'd measure with that would be metals. So essentially you send a sample through, it goes through a plasma, and it turns into this atomic form. So these individual atoms are going through there to look at the different metals in there. And then last but not least, of all things, MALDI is being employed in most microbiology labs. I'm not going to talk anything about MALDI, I'll leave that to the micro lab in the future. But so there's a plethora of things, and there's reasons to analyze different samples for different sources. In terms of mass spectrometers, we can break this down into essentially two forms. There's a nominal mass and an accurate mass system. So the nominal masses systems are the single quadrupole and the triple quadrupoles, and the accurate mass are the time of flight systems and the ion trap systems. Clinically, single quads and triple quads are probably the most common in the clinical lab. So if you heard of JSeegmiller, so that's a single quadrupole, and then LCMS, MS, that's a triple quadrupole. But however, accurate mass is starting to become more integrated into these labs as well too. It offers some advantages in certain instances. So a lot of the questions I get, people come up to me, they're like, well, Jesse, what mass spec should I choose? And so it really depends on your application. Do you want to look at a metal? Do you want to look at a protein? Do you want to look at steroids? What do you want to look at? So I just want to break down a couple examples. I don't want to focus too much on this. But atmospheric pressure chemical ionization, so that just means that ionization happens at atmospheric pressure. It's not under a vacuum. So electrospray, which is one of the most commonly used techniques in the clinical lab for ionization. This ionization happens to occur in the liquid phase. Okay, so the ions are produced in the liquid phase. Where APCI, or atmospheric pressure chemical ionization, essentially they apply a current between the probe and a needle, and there's this electricity going around, and it ionizes the nitrogen and the oxygen in the source, and that nitrogen and oxygen in the source then collide with your ions of interest, or your molecules of interest in the gas phase, so it's a gas phase ionization. Okay, so I kind of tell you this, the different sources have a different mechanism of ionization, but again, I'm going to one-line this and say the common goal of any source is ionization, okay? So that's the goal of any of these sources, they have to produce ions. So here's just kind of a schematic which shows electrospray if we had a positive mode here. So as this droplet, you can see, comes out, before it comes out of the probe, it has to be an ion, so it gets blown up. So you see these positive ions, and what happens to that droplet as it pulls out, there's a potential applied from the probe to the front of the mass spec. And as that positive ion is drawn to that potential, you'll get this evaporation, so your solvent molecules will go away. And then just like when we were little kids, we used to put magnets to each other, the positive poles, and they'll repel one another. So these ions get closer and closer together. You get this ionic repulsion, you get the molecular form, and then the ions go in. So it's kind of a high-level overview of ionization and electrospray theory. So here's a single quad. So the function of a quadrupole system is to harness ions. So I thought in terms of Wikipedia actually does a good job explaining these things, but I did have to laugh. I did have to capture this picture here, a bunch of ions. And so now that I've went through medical training, I know how to classify that this quadrupole suffers from hyperionemia. So that's my one joke. Glad you guys laughed. I don't know how to hear crickets. But the function of the quadrupole system essentially is to harness these ions and keep these ions in, to carry them back to the detector. That's what they're doing. And so I'm going to touch briefly on quadrupole physics. It kind of scares people when you hear that sort of thing, but it's relatively, hopefully, explained in a straightforward manner. So you have these vertical and you have these horizontal poles. And these vertical and horizontal poles are going to be, the vertical poles are going to be the same potential, negative or positive, and the horizontal poles are going to be the same potential all the time. So the horizontal poles are always going to be opposite of the vertical poles, okay? So if I can illustrate this, if we have our ions and they make it in the quadrupole, so here's our ions here, and they're attracted to this blue pole here. So as the ions migrate towards that blue pole, it oscillates charge, so it goes from positive to negative, so it repels those ions and keeps them harnessed in the quadrupole system, okay? And there's a certain type of RF frequency that they can apply that'll oscillate, if you oscillate these quadrupoles at a certain frequency, it'll harness certain masses. So some smart physicists figured this out. If they apply these voltages and oscillate them at a certain frequency, they can keep masses inside there. And we really don't have to figure that out as users of the technology, but that's what it went into it. So here's the basic layout of a triple quad mass spec. So we talked about a single quad. So again, we have our ion source. Here's our first quadrupole. So this is where we can transmit and harness ions and select ions out. And then you have the second quadrupole. And the purpose of the second quadrupole in triple quads essentially is to give a fragmentation profile. So the second quadrupole can bring in hot gas and those ions that we made and we transmitted into the second quadrupole. The hot gas that's excited can collide with your ions and break them up. So instead of looking at just a mass, which can be non-specific, we can get a fragment of that mass and we get a more specific fragment, which can be analyzed here or scanned out in our third quadrupole. And finally, it's detected here. And I'll kind of go over some generalized schematics on this process. So how do we get the ions to go from the front of the mass spec to the back of the mass spec, right? We talked about how we're able to harness ions in and keep them in the quadrupole by oscillating the frequency of the poles. But I used the guy from the Price is Right, which I haven't watched in 20 years, so here's the cliffhanger. And so if you look, here's what we have here across the quadrupole system is a potential gradient. So if this guy's a positive ion, it's going to pull him up the mountain if we have a negative potential gradient. So he's going to be attracted to the negative potentials. So then he'll go through. So we don't have to think about that either. Manufacturers have thought of that. So if we had our quantum mechanical microscope and we're able to look down a quadrupole system to see what's happening during this time, keep in mind we have the circular type motion of the ions, and now we're pulling the ions through to the detector, and it would look something like this. So it sort of corkscrews through the quadrupole system. So this is a simplified method. There's all these different. If we have a hardcore mass spectrometrist here, they'd probably argue with me on this, but that's essentially what it'd be like. So here's kind of to discuss resolution. So most quadrupoles are nominal mass systems, and what does that mean? It means that they're able to resolve 1 mat or 0.7 AMU, and what that means, if you look at mass 100, so if we put 0.35 AMU on one side and 0.35 AMU on the other side, it'll collect masses in that range. Okay, so that's what that means. If we need to resolve something within that happens to lie in there, then we probably have to go to a different instrument that has more mass resolution. But this is what a nominal mass instrument will do. So a lot of times you'll hear people talk about MS only or LCMS, and what MS only means is there's no fragmentation. Okay, so if we send an ion through, this 821 ion, it goes through. Remember our Q2, this is where the fragmentation would occur. It's not fragmented, it's just transmitted through Q3 and detected out the back. So that's MS only in triple quad. So to show you an example, here's some a compound that I infused here at the university, lohexol. You don't really have to pay much attention to the structure, but down here is the chemical formula. So the chemical formula, I can then take this chemical formula, put it in a calculator, and it'll spit out this exact mass, 820.8803, right? So if I infuse this compound, though, experimentally I get 821. So why is this? Well, remember back to my first slide or whatever it was, a compound has to be charged in order to be analyzed by mass spec. So what's happening here, if we add 1 to this mass, we get exactly that, right? So we get 821.9. And so what's happening is a proton, or hydrogen, was added to this compound to give it M plus H, to give it this adduct, to give it that charge, to get it into the mass spectrometer. And I thought this was an interesting picture too, because the standard I had was from actually a contrast dye that we give patients. So there was residual sodium left around. So if we add sodium to this, 23 to this, we get 843, which is exactly what we see here. So there's a mixture of that. So you want to minimize the different adducts if you're going to look at different compounds, for quantitative analysis. But it just kind of shows you that you can have all sorts of different adducts: sodium, lithium, hydrogen, and you can use those. Ammonium adducts are used for therapeutic drug monitoring, those types of things. So if you ever hear of somebody talking about an adduct, that's what that means. So now we're going to go to MS. So instead of letting everything through and scanning, now we're going to put our blinders on. We're only going to let this mass through here. Okay, this 821.8. We're going to hit it with a hot gas in the Q2, and it's going to produce these ions. And then we're going to get what's called a mass fingerprint. So here's a product ion spectrum, and I highlighted a couple different product ions. So these are the ones that I use. This here is a loss of 18, and generally when you look at methods, if you have a loss of 18, that's a kind of a hydroxyl loss. So you lose, they'll talk about a water loss. So that proton that we put on, that guy goes away along with your hydroxyl group. And so lots of compounds have hydroxyls on them, right? So that's considered a non-specific sort of fragment. But somebody published on it, so I keep it in there for academic reasons in my method. So this can be considered a mass fingerprint. So we can take this mass fingerprint, put it into a database, and then we can search this mass fingerprint. So this could be a protein, a small molecule, or whatever. So generally what we're going for in the clinical lab is we want to just look at one thing. We don't want to be scanning all sorts of things and picking things out. So we go to this very focused. We put the blinders on, we let one thing go. So we turn the channel in the first quadrupole to 821, and we turn the channel in the third quadrupole to 656, and then we only let those things through. So if anything, nothing else, no other masses can get through beside those two at that specific time. And then later on, we turn our channels over to the 614. We then hit that with the hammer, break it up, and then we're looking at the 486. So this is how we're able to multiplex and do quantitative analysis on multiple markers. So there's also an untargeted approach. So that was a targeted approach. So we're very focused. We only let things through at a certain time. So an untargeted approach would say, okay, tell me what I need to be looking at. And so usually what you'll do is like an MS-only survey. If that survey scan hits a certain criteria, which is usually refers to an intensity threshold, so we set this if-then statement. So if the intensity reaches X counts, then perform the product ion spectrum. Okay? And then we can get the mass fingerprints of these compounds, and this can happen multiple iterations throughout your method that you choose. So this is referred to as sort of a data-dependent approach. Okay? So whatever goes in as high abundance, we're going to target that, get the mass fingerprints. We can load that information up into a database. Okay, so how can this technology help us? So in the clinical lab, many of the testing modalities we have rely on some sort of optical detection. And so some of these techniques are prone to interferences that also absorb at the same wavelengths of our assays. Okay, so I just kind of highlighted here hemolysis, icterus, and lipemia. So the HIL indices is a hamper on our some of our clinical tests. So if they reach a certain indice level, we cannot perform that test reliably on that species. So mass spec, the nice thing about mass spec is it's not influenced by optical absorption. So just to kind of take you through a couple different approaches. So from a small molecule approach, generally what people will do is they'll remove the proteins. They use some sort of organic solvent or acid. They pellet the proteins and centrifuge them, and then they inject. And this is referred to as a crash and shoot or a dilute and shoot. So this is ideal. So about 90% of the methods that are in place are probably follow this method. If you need to get cute, then you can do some other preparatory up front. So I used this dilute and shoot method for the lothalamate study that I did down at Mayo. And lothalamate is a compound, a radiocontrast dye, that is supposed to be a perfect marker of GFR. So a perfect marker of GFR means that if it passes into the glomerulus and it's filtered by the glomerulus into the proximal tubule, it stays in the proximal tubule and is not reabsorbed into the peritubular capillaries, okay, and it passes into the collecting duct. Okay. If it's not filtered by the glomerulus and passes through the peritubular capillaries, it cannot be secreted, so go from the peritubular capillaries into the proximal tubule, because that would not be a perfect marker of glomerular filtration, right? So they have to pass back into circulation. Another criteria of GFR markers, it cannot be metabolized upstream either, or excreted some other way. It has to be excreted through the kidney only. And so why I highlight this test is because when I was doing this study, we came across a sample with interfering substance. So here was our typical CE UV absorption electropherogram, and here's a typical lothalamate calibrator. Okay, we came across this patient that had some sort of interfering substance present here, and if they calculated the GFR, it was non-physiologic, right? 314 mils per minute. And we simultaneously would always get creatinine clearance as well, and so creatinine clearance estimated about 31 mils per minute. So I tried this sample on the mass spec, and I had 4 different transitions that I monitored throughout the method. And there's the 3 in gray that are around 20, lower 20s, and this one in red here I highlighted goes up to 32. But this one here, this 456 transition, you can see there's no other chromatographic interference. It's a single peak, a nice looking peak. So there was no interference with those. But the 336 is shown here. So prior to giving these contrast dye injections, we collect a urine sample. And we collect a urine sample to see whether or not there's an interference present the patient has an interference present prior to giving this contrast dye. So here's the 456 transition, and this is just noise here. Okay, so this is like 20, 30 counts. This over here is 700,000 counts. Okay, so whatever this patient had a very similar mass to lothalamate, and it had a very similar fragmentation profile to this one. And I published this because I wanted people to be aware that this transition is the most sensitive. If we do it from a pure analytical chemistry point of view, we would use this transition. Okay, so I wanted to warn people that not to use that transition. So we're still able to look at other more unique fragments in order to get the correct result in this case. So it just kind of highlights, not every single MS-MS transition is going to work for you. You may have to find other ones that are more focused on what you want to work for. So this just kind of shows creatinine creatinine clearance overestimates GFR with respect to lothalamate clearance, which is well known. And then early this year, 6 years after I did all the work, we published this data where we compared lothalamate and lohexol. Okay, and so the reason why we did this study is because we wanted to at Mayo Clinic they didn't have a plasma disappearance protocol, and so we wanted to prove that lothalamate and lohexol were the same, and so we could monitor lothalamate while we were monitoring the lohexol plasma disappearance. So if we proved equivalence, we could move forward with this, but much to our chagrin, we saw about a 15% difference between these two in terms of renal clearance. And then over here, I just kind of show the creatinine. So again, we saw 20% in the previous study. Again, I see 25% here. This is a much larger study, much more power. And then lohexol just add 15% to the top of that. So the bias was consistent, so we're trying to figure this out. So after one of the criticisms of this study, through the review process is, we didn't give enough contrast dye. We didn't give a high enough dose. The reason why we gave a 0.5 ml of the contrast dye is lothalamate was validated against inulin, which is considered the gold standard for GFR measurement at that dose. And so typical plasma disappearance methods, at least in the US, give about 5 mils of contrast. So maybe the dose has something to play with this difference. We're not sure, so it would be a 10 times higher dose as opposed to what we looked at. So actually, we're proposing a new study here, and I have a method set up to do lothalamate and lohexol. We want to look at 20 patients where we simultaneously look at renal clearance and plasma disappearance to kind of tease the differences out between these markers. Okay, so now I'm going to switch gears a little bit to, I think, which is a really hot topic, opiates. So opiates are a clinical nightmare. You can get to morphine through an illicit drug called heroin. So heroin can metabolize down into morphine. If you give codeine, codeine can metabolize into morphine. If you eat poppy seeds, they can metabolize into morphine. So there's all these different things that you have to think about when you look at monitoring opiates. Another thing I want to highlight too is there's morphine can metabolize into hydromorphone, and codeine can metabolize into hydrocodone. And these things happen to be isomers. Okay? So I showed a chemical structure here. I promise these are the last chemical structures I'll show you guys. But here's codeine and hydrocodone. And if I I take the molecular formula, I put it in my calculator, I get this mass of 299.1521. And the formula happens to be exactly the same for hydrocodone, with the exact same mass, right? So the differences between these two is codeine has this hydroxyl group here, so there's a proton here and a proton there, and this unsaturated double bond. It turns into a ketone, and now this double bond here is saturated with those two protons. So that's the difference between the two, if you really care about the chemistry. But the problem is, from a mass spec point of view, is we can't separate them by mass. And another problem, if we do MS-MS to get more specific and look at specific fragments, they have the same fragmentation profile, or a similar fragmentation profile. So those fragments, we can't separate those, okay? For those of you, so I didn't want to scare all of you, so I decided to illustrate this through this young mite hockey player, okay? So this young mite hockey player has his jersey on the right side, the right way, and then he just picked that same jersey and flipped it around. So he has the exact same mass, right? So he's just an isomer.
Is that your kid?
Yeah, this is my son. And I've named him appropriately for the opiates, Trentone and Trentine. And then if he goes through the Mike Gong show and it produces some fragmentation pattern, he gets his jersey pulled off from the game, this jersey is going to weigh the same in either case, right? And then the kid stays the same in either case. So that's one way to kind of think about these problematic things. So we can't separate it by mass, we can't separate it by MS-MS. So then we have to employ, when you hear LCMS or LCMS, this is why LC is not going to go away anytime shortly. So LCMS or HPLC is high performance liquid chromatography, and you get your mixture of analytes, and it goes into a stationary phase, which is noted here, these little red dots. The mobile phase carries it onto there, and it depends on the affinity for that stationary phase. and the migration rate, the partitioning in and out of the mobile phase, and which will depend on the elution time. So this is better seen here. So we have our mixture, okay, and as it migrates through the column, hopefully they're able to separate by partitioning in and out of the solid phase and the mobile phases, and they'll do it at different times. So the opiates, we need to do this sort of thing with that, and that schematic I showed is really simplified actually too. It's much more complicated than that. All right, so let's switch it up again to proteomics. So I just want to give, again, general approaches. You get your sample, you digest your proteins, the peptides. Typically, when you look at the literature, they're going to be using trypsin, and then you analyze the specific peptides by an LCMS MS method. So you can do this either qualitatively, where you have an untargeted method, and then you submit it to a database, and you search the database, and it'll tell you which proteins there are. And then you can do it quantitatively, where you look at a specific peptide, and you refer that peptide to a calibration curve, and to get a quantitative result for your protein. And I want to highlight some light chain work that we've done here, and also that's been done in other places. So Dr. Murray, actually down at Mayo Clinic, was interested in looking at monoclonal gammopathies of immunoglobulins. And so he wasn't happy with the traditional techniques of immunofixation and serum protein electrophoresis, so he decided he wanted to look at mass spec of these things. So what he did is he took the sample and he purified the immunoglobulins. We talked about how we generally digest these things, but what he did instead of digesting it, he just did a reduction step. So he did the reduction step, he removed the light chain from the heavy chain, and therefore the light chain was then intact, and then they shot this on a mass spectrometer. And kind of the really cool figure, and I thought the cool takeaway from the study is a great personalized lab medicine story. So here's our traditional ways to look at these M spikes, or monoclonal proteins. As you can see, this M spike initially when the patient came in. So this is a patient they followed over 7 years. And the M spike that when they came in was positive. It was high, and the immunofixation was positive. And over here is the light chain result. Okay, so he's able to look at this mass over this course of 7 years and stay with this mass over this course of 7 years. And they can also watch the therapy, so they can watch the relative abundance of this this light chain go down and up through the therapeutic changes in this patient's treatment. I thought this was really cool from a personalized medicine approach. You can follow this exact same light chain over the course of therapy. However, as a future laboratory director, patient-to-patient monoclonals are going to have different masses. Okay, so this is going to be, we have this moving target between patients. Okay, so one patient's monoclonal is going to be just slightly different than this other patient's monoclonal, which is slightly different. So, this is a great story, but how do we put this into a lab and for routine use instead of having a mass spectrometrist analyze these patient samples? So, begs the question: is this too focused for a routine lab? If we had a mass spec for every myeloma patient, that'd be great. So Dr. Carter was interested in looking at urinary free light chains. So kind of the story behind that is there's a literature debate about the accuracy of the free light assay, which is a nephelometric assay. So I was like, okay, well, let's try a different approach. Let's try and digest them up, the general protocol, and see if we didn't have an accurate mass system available to us. So let's see what happens. So I took the free light kit assay controls, and they have a Kappa and a Lambda control, and I developed the untargeted MS method. So again, if a mass reaches a certain intensity threshold, then it's going to perform the product ion scan and get the mass fingerprints. And then I can take these results and submit them to a database. So here's my database results. So I submitted to MassCap. This was a freeware. So I did this as cheap as we could. The one thing I want to highlight, so you get your protein hits down here. So there's like albumin, transferrin, other high abundant serum proteins. The one thing you won't see here is the Kappa light chain. So this is the Kappa results. I don't see Kappa light chain, and this is the Kappa control. I did the same thing for Lambda. Again, I see high abundant serum proteins, but I don't see a hit for the Lambda light chain either. So this is pretty surprising because how is this assay even working, right? So the general search results, there's no database hits for either Lambda or Kappa light chains using this non-targeted approach. And so I knew from the hits, though, that this sample had been digested by trypsin. And there's other proteins. It's a much more complex mixture than just a light chain mixture. Right? So then I switched it over. I went to a theoretical approach, which I never do. This is the first time I've ever done anything like this. I generally do brute force, where I get the protein of interest and digest it up and let the data lead me. But I didn't have that luxury in this case. So I used a software program called Skyline. And what Skyline does is it takes your protein sequence in, it'll digest it up and recommend different transitions for you to put in your methods. So I figured, well, I only have a couple weeks before an abstract submission, so what do I have to lose? So here's kind of what the data from Skyline gave us. So here's the Kappa. So these light chains, we just looked at the constant region in these light chains, so they're very small. So we don't have a lot to work with either in this protein. It's not a big protein. And then there's the Lambda sequence. So Kappa only has one clone that's known as of right now. The Lambda sequence has 5 different isotypes. So there's some redundant, there's a lot of homology between the different Lambda isotypes. So then I just condensed those duplicate peptides in there and came up with a method for Kappa and Lambda. So also too, what these databases and all these things won't tell us is we still have to look up the informatics and what's happening, what's the biology behind these peptides and proteins. Is it modified? Is there a mutation that we need to be aware of? Is there a post-translational modification? So you have to look at the informatics behind these things, and then I just make sort of informatic notes. Is it unique to this protein, etc. Okay? So we don't want to like develop something that's non-specific and then get to the end of that, start from assay development or assay validation and find out we're not specific enough. So this is just kind of shows some of the different work that you need to put in. So with the Lambda, there's this IG Lambda-like polypeptide 1 and 5. Is that of significance here? We're not sure, but I do have it noted just so we know when we're not caught off guard. Should we go forward with this project? So I took that data from Skyline and then I did the MRM. So remember MRM, I'm just focused. Before I was scanning, whatever was high abundance is going to give me my data. So now I'm just focused, right? I just took that Skyline data and now I see these peptides popping out. And actually a pretty good signal too in the control. So if I look at the same control, here's the Kappa control, the peptides are in there. Here's the Lambda control, the peptides are in there. Okay? Here's another example. So this was a common peptide as well too. So kind of like my take-home message from this project, those peptides, the Lambda and Kappa chains were in there. The light chains were in there. The MRM system was much more sensitive and much more focused than the general screening in this case. The initial data appeared to be promising for the signals of the transitions, but really it highlights the difference between the untargeted approach versus the targeted approach. Okay? So if you take a sample and it has some low-level protein in it, you give it to the proteomics core, they might not see that low level if there's 5 other things or 10 other things that are much more abundant than that. So that's kind of the take-home message I'm going to point here for that. And also to keep in mind and consider plasma proteins. If we look at the plasma proteome, I thought this was kind of a nice schematic. Actually, this is for a product that removes the high abundant plasma proteins. But it does a good job at showing that 90% of the plasma proteins consist of albumin, immunoglobulins, transferrin, etc. And then of that 10% that's left over, it's broken down essentially into complement and apolipoproteins. Okay? And then you're fighting for this 1% here. Okay? So if you're down, if your biomarker is down here in this 1%, and you don't do some sort of preparation up front, you're not going to see this thing, right? I guess that's my take-home message. And keep this in the back of your mind. So now I'm going to switch and kind of contradict myself for the untargeted approach, not really though, because I'm going to talk about the amyloidosis work that's been done down at Mayo Clinic. So the amyloidosis, what they do is they'll get these tissues and they'll stain it with a Congo red, which is shown here. And they perform this laser microdissection. So they put this on a special slide and they have this red beacon here of the amyloid plaque. And the special slide, what happens with the laser is the laser hits the back of this slide and it puckers. It puckers the tissue off, and the tissue is collected into a vial down below. And then all they do is just what I did before with my light chain digest. They digest the plaque up, they submit it to a database. So here's the results over here. So you get the different hits, similar to like what I had. They actually use Mascot search engine. There's 4 different search engines they use. But what it showed, that for the initial diagnosis, which here's a Kappa chain, the mass spec correctly identified essentially every case of the different amyloidosis classification. And this is just, there's no quantitative analysis here. This is just through database searching. So if you're specific in the sample that you're looking at, you might be able to see these high abundance proteins. So I've seen it before with the molecular tox where you have non-cancerous tissue diluting out your cancerous tissue. You know, this approach too may be good for proteomics, and that might be help you see what you want to see instead of just digesting up a non-specific sample. So if you get specific, you can maybe tease out these information. And you as pathologists are going to know where this technique may work best. As chemists, we just don't have the knowledge base. So this is really exciting too. So there's a mass spec clinical analyzer that is being released. I saw this at the conference I went to the end of February. And so one of the problems with mass spec is it's not automated enough. There's a bunch of user intervention. The sample prep has to be done manually, or it has to be carried over into a robotic system. But there's manual intervention that has to be input. What this company did is they put essentially like a chemistry analyzer or an immunoassay system front-end preparation up front. So the samples, you can load them directly on, and then the samples aliquoted and prepared, reagents are added, and then it goes to the HPLC system, and then from the HPLC system it can go to the mass spec and be detected. So this is much more amenable to a clinical type workflow. So automated online sample preparation, so you get your results on demand. And so kind of areas I think that this could make a really big impact. You know, last Friday we had busulfan analysis where the pharmacy is looking at the levels of busulfan and whether the patient's doing good or bad depends on that busulfan result. We have to send them all the way over from West Bank, and a couple hours later, they get their results back. You know, other things too. I think anesthesiologists, you can get more on-demand result. Look at the pharmacokinetics in your patient more real time rather than waiting a couple hours for it to go to the lab, go through the standard processing. So if you had a system set up, you could put it there towards these procedural rooms and then analyze your samples within a few, 10 minutes or so. This is really cool. Hopefully you guys enjoy this. There's another system that came out at this talk. It's been out for a couple of years now. The rapid evaporation ionization mass spectrometry system. So there's a little video of what it is. I couldn't block out the water's thing. But here it is, this iKnife, okay? So the iKnife is this electrosurgical system. And what this electrosurgical system does is it goes through and it cuts tissue. Okay, so it's showing the cut tissue. And then the tissue vapor is then sucked up through and into the mass spectrometer. Okay. And then the same principles I've been talking about initially in my first part of the talk, apply. So it becomes ionized, and then it evaluates the different masses. So here's the ionization. There's some marketing in here too. There's some marketing to get rid of the stuff we don't want. Each vendor has their own way to do these things. So it goes through, and then it flies on. So here's the TOF, a time of flight. So it's flying down the tube and being detected. So the important thing here is you're getting this chemical profile real time of your tissue. Okay? And so I saw this talk by this Dr. Senjin out of the UK, where he's looking at real-time classification of breast tissue. Okay? So he used the cut and the coag modularities, and he showed that he got great sensitivity and specificity when looking at these breast cancer tissues in sort of a gross room. And I asked him for his video because it showed him cutting across the tissue, and he'd say, now I'm in tumor, and then the screen would flash red. And then he'd go over to normal tissue, and it'd flash green. Okay? And he didn't send me the video, unfortunately. But there is a video out there. If you search this on YouTube, there's a video from a couple years ago that will kind of show you what that would sort of mean. there's no chemistry discussed in this talk, but what they did is they classified the different breast cancers, put those into a database, those chemical signatures, and then they compared those real time. Okay? And what I gleaned from there was a lipid profile, which I have to assume is from these cell membranes, that's what they're classifying these things as, because they can take this system and go to fish and tell the difference between fish species, the difference between horse meat and cow meat, those sorts of things, right? So, there may be other areas that we could maybe employ this in and do some research. If we got this in a gross lab, maybe we could find out some more interesting things here. But I think this is really a cool technique, and hopefully you guys do as well too. I'm doing good on time. So in conclusion, you should be able to recall some of the basic concepts of mass spectrometry. You have to be an ion in order to be analyzed by mass spec. You should recognize some of the challenges and different opportunities within the different approaches you take. A small molecule approach versus a proteomic approach. So small molecule, I showed that we're able to remove interference by mass spec, but we also have this issue of isomers you have to keep in the back of your mind. Hopefully I've been able to spark you guys' interest and identify potential areas of integration for this technology as well too. Oh, another thing I want to highlight too is the general survey, the general survey approach. When we looked at the complex mixture of the buffer, we didn't get anywhere. But when they look at the amyloid plaque and they look at the general survey, they see what they want. Well, they're looking exactly at what they want. So that's going to tell them more information, right? So we get more specific sample, or we get more specific information versus a targeted approach. So hopefully you're able to take what I've talked about today and see some areas where you can potentially integrate this technology into your research or into your lab. The on-demand drug sample analysis for the anesthesiologist, the busulfan analysis for the pharmacy, I think that would be a great place for the clinical analyzer to help them out. And then again, the real-time tissue analysis. I didn't know how much time I'd have, but there's another thing that I want to talk about. So actually, the University of Minnesota has some pretty deep mass spec roots. There's a Dr. Nair. So Dr. Nair came up with this isotope ratio mass spectrometer. And his group back in the late 1930s was the only one capable of analyzing uranium isotopes. And the reason why that was important during that time is because of the Manhattan Project. So what people would do from all over, they had Oak Ridge and Berkeley that were trying to make and separate these uranium isotopes. They would send him samples, and he would be like sort of the QC lab for the uranium. So it's kind of interesting. And I knew about this about 10 years ago, back before I had kids, and I had a lot of time, and I read this book called The Making of the Atomic Bomb, and they discussed Dr. Nair in this book. So I thought that was really interesting. So with that, I will take any questions.
Thank you for the very interesting presentation. The video that you showed towards the end, I saw that in relation to the surgical margin. When the surgeon is cutting, you can tell if the margin is negative or positive. You don't need a frozen section then. This field that you presented today probably represents a paradigm shift for anatomic pathologists, because the technology you talked about does not involve preservation of tissue. Actually, it involves destruction of the tissue to render the result. And then anatomic pathologists need to evolve into understanding the purpose of the sample as to produce results that is meaningful to the patient. It will take time for all regulatory bodies to acknowledge that, and we see it. There is a lot of push towards in this direction to get us out of the mindset of preserving tissue and that sort of thing. But this is very interesting.
Yeah, I think early adoption of this sort of technology would probably be best suited in a gross lab. The Dr. Senjin, he's going to take this into the actual OR. This sounded like this spring. according to his talk, whether he does that or not. But most of this stuff is being done in the growth lab, so maybe that would be help with that. You know, where you don't you have the real time, which that's where you want to get to, right? But you're right, you're going to have to take time to convince people that this is adequate, because it will be a paradigm shift in your practice.
Nice talk. I'm not very familiar with mass spec, so it's always good to have it reinforced. So it's really neat technology, and you have a lot of expertise in it. So how do you think, what would be your recommended approach for clinical lab testing? So if someone presents a problem to you, your choices are send it to a reference lab, develop a mass spec test in-house, versus use another platform such as an immunoassay. How do you decide what's best in an individual situation? What guidance would you recommend?
Yeah, so generally what I think we usually will guide us to that answer is how good the test is. If the test isn't good, and the reference lab has that test, and depending on volume too, if we only get 2 of these a year, it might be better sent out rather than keeping that test out. But if we get significant volume, that's something we want to bring in, because we lose that revenue. So whether or not we can do that, you know, do we have the resources to do that? You have to consider that as well too. Do we have the instrumentation to do that as well? So those are the types of things. But I think in term, you know, if you have an idea where you would like to try this technique with, the Cancer Center has a microdissection system. Okay? And then we could use the Proteomics Center, use their system that's set up to do the proteomics analysis to kind of look at these sorts of things. So kind of a proof of concept before we dive in and make a $2 million investment on something, typically for like a pathology. I'm thinking in terms of pathology now.
Just a comment pertaining to that, Jesse. Bartek, is that you? Oh yeah. Do you want to comment on what you're looking into with these?
So that's the things I would like to comment. First, that there is an instrument already on campus in the mass spec facility in the chemistry, which is capable of what they call MALDI-IMS, multi-image mass spectrometry.
Yep.
And the technology that I kind of explored recently is a combination of that with staining with antibodies coupled to unique metal ions, which would enable sort of similar thing to immunohistochemistry, except you could analyze 50 parameters at the same time. But the resolution of those methods is a little bit an issue still. Right. But I was wondering if you have any information about the instrument which is already on campus.
I don't have anything.
SIX 5700.
Yeah, I haven't worked directly with a MALDI imaging system. The one thing about MALDI though, and you said MS, right?
Yes.
MALDI MS is going to, whatever proteins are in there are going to go off intact, essentially. Okay. So if it's an intact protein that has modifications versus a protein that doesn't have modifications, the same protein, those are things that you have to sort through. And what does that mean? Those sorts of things as well.
Would the free database that you mentioned two. One was Mascot and the other one was the one that you used to zero.
Oh, Skyline. Skyline is just
Skyline.
Yeah, Skyline's not a database. That's just essentially.
Would that be able to predict, let's say, parameters for something like, let's say, collagen type 3?
Absolutely. If you want to digest it, so that's when you go and digest. So the purpose of digest, some people don't want to do it, but the purpose to digest a protein, let's say if there is a glycosylation that's different amongst all the proteins, right? We probably wouldn't want to focus on that end of the protein. We probably want to get into a unique part of the protein that doesn't have a post-translational modification and then focus on that guy. So that's why we digest them up. And then also, too, internal standards are easier to get in smaller locations.
Is the issue of analysis of image, like tissue in a 2D structure, is it possible to combine that with digestion?
I mean, that's kind of like what the microdissection sort of thing would do, right? I guess it depends. You can take either approach. If you can get the answer that you want from a MALDI system, that's great. If that gives you the information you need.
What you're saying is MALDI itself does not involve digestion.
Right.
Okay.
So that would be like a more of a real-time type thing. You don't have to go through that process. But if it's not specific enough for you, or that you see these masses going all over and it doesn't quite make sense because there's different modifications. Again, I don't know the proteins exactly, but you have to look into that and see if that's going to be a problem for your analysis. Yes, Dr. Berry.
Just an FYI, we've been using for a few years MALDI-TOF mass spec in the microbiology lab. It's been really transformative in terms of rapid identification, moving workflow through, and for certain groups of microbes in particular. But as you point out, there are protein spectra that are identical for some organisms, so we then have to rely on a phenotypic quick test or some other biochemical physiological profile. But ours is based on the Shimadzu mass spec technology.
Mm-hmm.
The competitor is the Bruker system. And we evaluated the research use only database before FDA then approved a database. And because we did the RUO, then we have free access to it. Other people have had to purchase the use of the RUO database. But I like it a lot, but you still need to be a really outstanding commander of phenotypic microbiology, where there are labs that take out, any answer comes out, they report it. It's quite wrong.
Yeah, you definitely have to know the technology and know the weaknesses, and that's why I try to point out weaknesses in different types of sample and different workflows.
What I tell our residents is, on the board exam, it's not going to be, the answer is not identified by MALDI-TOF mass spec. And so, and our new techs in the lab, they're not learning traditional microbiology now. They'll, on lab rounds, would say, you know, yes, we identified by MALDI, and the residents on rounds from clinical services have no idea what we're even talking about. So it's a great concern by many people in the field that we're going to be losing the art of microbiology.
Jesse, I just wanted to expand a little bit on some of this discussion. You mentioned at the beginning of your talk that basically there's nothing using mass spec approved by the FDA. Could you expand a little bit about how we go about getting FDA approval for mass spec technology?
Right, and that's kind of one of the reasons why I'm in fellowship. The FDA came a couple years ago to a mass spec vendor who I used to work for, and they said, you know what, you can't sell to clinical market if they're going to use this for some sort of diagnostic, as a diagnostic tool. Okay, so the only mass spec that's FDA approved is the MALDI system that I'm aware of. So they're making inroads to manufacture. It's this big bureaucratic, long process to make sure the manufacturing is in line with what the FDA considers appropriate. So then from that point, then you got to go through just like a 510K process with any assay that you're going to do. The problem with doing that, and I think the linchpin of LCMS MS, is that all these compounds and all these columns can kind of be variable. So the manufacturing process of the columns and the LC process and the LC systems is not quite there yet, as opposed to like a chemistry analyzer where you just subtract the background signal of the cuvette and then you're good to go. So mass spec isn't quite there, and these systems are just a little bit different. Each system maybe just a little bit different than the other. So I think that's the hang-up. Not that it's not a superior technology, but that's the problem, I think, getting these things through the FDA.
Okay.
And you have to take, and a manufacturer, as long as they're selling, they have no incentive to go forward with this, right? So I think the FDA is kind of putting pressure on to move in the right direction.
Yeah, we'll take last question from.
Oh, I just had a quick one just about derivatization. I mean, years ago, you used to have to derivatize. I guess it was more for GC mass spec.
Yeah.
You didn't touch on derivatization. It kind of faded as a...
Yeah, so with LC, the nice thing about LC is you don't have to derivatize. So with the GC, you have to put it in the gas phase. So as long as your compound's in the liquid phase, you don't have to derivatize it. So that's one nice thing about, that's why a lot of labs are moving from GC to LC. So that's why I kind of talked about LC.
All right.
Thank you.
Thank you, Jesse, for one.